Okay, the last topics in the chapter to talk about are genetic strategies. The first is the concept and the phenomenon that for every protein in an organism, there are often more than one type of that protein or more than one variation of that protein. Hemoglobin is a good case in point. Turns out that as a fetus, we make different hemoglobin than we do as an adult. And some people in Africa and Asia make yet a different kind of hemoglobin to withstand malaria. Fetal hemoglobin consists of four proteins like adult hemoglobin, except the four proteins are two alpha proteins and two gamma proteins. Fetal hemoglobin, as a result of that, has a higher affinity for oxygen, which means it has a higher attraction to oxygen, or oxygen has a higher attraction to it, and it's able to work in the fetal environment better. Adult hemoglobin has two alpha and two beta units, or two alpha and two delta units. There's different kinds of adult hemoglobin also. When mom's blood gets into the uterine wall and the fetal blood gets next to it, the fetal hemoglobin can pull oxygen off of the maternal hemoglobin and then carry it to the fetus's body, the fetal body cells. That is a great adaptation for making sure that the fetus can actually survive in an environment where it's not really exposed to outside air. It has to pick up its oxygen from maternal blood. Sickle cell anemics make different kinds of hemoglobin to survive infection by malaria. So in different parts of the world and in different phases of human life, we make different kinds of hemoglobin as adaptations for different strategies of survival. What these graphs are talking about here, this is hemoglobin, chains of hemoglobin. Alpha is in red, beta is in blue. The heme is these little green sections here, here, there, and there. Those are the parts that actually carry the oxygen. In a baby, the blue beta units are replaced with gamma units. So you can see in this graph, babies make lots of alpha and lots of gamma proteins to make basically fetal hemoglobin. Adults make lots of alpha and lots of beta, a little bit of delta. Most adult hemoglobin consists of two alpha and two beta proteins. Most fetal hemoglobin consists of two alpha and two gamma proteins. And this is a great adaptation for a baby to live off of maternal oxygen supplies. So, to talk about this more in depth, we have to take a look at what is called hemoglobin saturation curves. So, oxygen binds to hemoglobin easily, but it also releases, hemoglobin also releases easily from, oxygen binds to hemoglobin easily, but oxygen also releases from hemoglobin easily. The graphical relationship for the oxygen and hemoglobin is right here, and it's called a sigmoid curve. What this shows is that at low oxygen pressures, hemoglobin pretty steeply is attracted to the oxygen and picks it up. But when each hemoglobin molecule can carry four oxygens. Once a hemoglobin molecule has three oxygens, it actually doesn't grab the fourth oxygen as aggressively. And as a result, we describe this with this part of the graph flattening out here. So hemoglobin molecules do not grasp oxygen in a linear fashion. The if a hemoglobin molecule is empty, it aggressively grabs the first oxygen, the second, and the third, but not the fourth. pH can affect the position of this curve and the sigmoid relationship. If pH falls, the curve flattens out, and what that means is that oxygen loses the ability to hold hemoglobin. So the higher the curve, the more oxygen the hemoglobin is able to hold, the higher its attraction for oxygen. The lower the pH, the lower hemoglobin's attraction for oxygen. This is a great adaptation for how hemoglobin has to work in the human body. The pH of lung cells is about 7.4. When the blood comes into the lungs, The higher pH in the lungs allows hemoglobin to grab more oxygen in the lungs here through here than it would grasp if the lungs had a pH of 7.2. When the blood gets into the body capillaries, the pH there falls because of the carbon dioxide coming in from the Krebs cycle, dumping carbon dioxide into the cytoplasm, which goes into the blood. At the lower pH, hemoglobin cannot hold as much oxygen. So what happens is the oxygen pressure in body capillaries is down here around 40. Well, the hemoglobin can't hold because the pH is lower. The hemoglobin can't hold on to all the oxygen it picked up in the lungs, so it drops more oxygen off at the body cells because of the pH shift. So at the body cells, pH is low. In the lungs, pH is high. The high lung pH makes hemoglobin more able to pick up oxygen in the lungs. The low pH in the body cells makes hemoglobin more capable of dropping oxygen off at the body capillaries. So it's a great way for a molecule to oxygenate body tissue. So now the last topics to talk about here is gene duplication. As it turns out, we have more than one copy of many genes in the body. First off, to be heterozygous is a good thing usually because you can make two different proteins at least, which gives you a better chance of surviving in a condition that's not stable. So a heterozygote is generally more advantageous than a homozygote under particular conditions where conditions are changing. The organism basically can make two forms of a protein, which gives it functional resilience in response to environmental stresses. Hybrid corn, for example, is a much more vigorous grower than either of the homozygous plants from which it was made. Mutts, which are like hybrid dogs, are generally much healthier than their purebred or pedigree counterparts. Heterozygote advantage is important to evolutionary fitness. The sickle cell gene is a good example of how heterozygotes are advantaged. In Africa, sickle cell anemia is an issue because malaria is an issue. So the area in orange here, in pink and orange, is there because the malaria parasite resides there in the mosquito population. And there, the people that are heterozygous for sickle cell anemia have a better chance of surviving and growing into adulthood. And as a result, they pass that gene down to their offspring. Now, if a person is HB, or H, I should just let's erase this and I'll write it over again. H is the hemoglobin gene. HA means that you're making homozygous normal alpha proteins for your hemoglobin. Everybody makes the beta proteins, but it's the alpha units that are affected in the sickle cell trait. These folks die of malaria. If you are HAHS, you don't transport oxygen quite as well, but you survive malaria. So this person actually survives malaria. If you're HSHS, you're probably going to die of sickle cell anemia because your blood clots so commonly in your bloodstream. So let's scroll here and take a look at this. These are what sickle cells, we've seen this in a previous unit. When let's scroll this back a little bit. When blood cells are made of normal hemoglobin, the red blood cells go through the capillaries in their normal shape. This is called a biconcave disc, and these little discs shoot through the capillaries and they flow very nicely. When the hemoglobin is abnormal, instead of two alpha proteins and two beta proteins, it's got two sickle proteins and two beta proteins. The red blood cells make these little goofy stringy shapes. They don't go through capillaries well and they get stuck and they form blood clots. If a person is a carrier of sickle cell anemia, pretty much their blood cells are shaped like this unless they get extremely hypoxic. But the malaria parasite cannot successfully reproduce well inside of their cells, and as a result, they have a resistance to the parasite that causes malaria. Now, One last topic to consider in this chapter is gene duplication. A major aspect of vertebrate advantage is gene duplication. Due to some probably accident of meiosis about 550 million years ago, genes for the formation of the head got accidentally duplicated in some ancient vertebrate. And with those extra genes, vertebrates could grow bigger heads with bigger brains and bigger mouths to eat bigger amounts of food and grow to larger sizes. That accidental mutation resulted in the rise of a massively dominant group of life forms on Earth. The relatives of those ancient vertebrates that didn't get that gene duplication still live in the oceans today, and they're no bigger than a human finger. They're very, very diminutive, very, very small. The nice thing about gene duplication is it creates a situation where there are more than one copy of the gene. What this does is two things. First, the organism can make more of the protein. Secondly, the extra genes can actually mutate into to make other proteins, which could give other capabilities for the organism. So human hemoglobin is a case in point here. Multiple copies of the hemoglobin gene have allowed humans to make basically 4 different polypeptide chains for the different kinds of hemoglobin. And we talked about that. The alpha, beta, gamma, and delta. All 4 of those give us more capabilities than only one would. So humans also carry a non-functional hemoglobin gene that is silent because the promoter is damaged. We all carry it. It just doesn't work. So that's evidence that maybe at one time we made a 5th kind of hemoglobin. Fish in cold environments make all kinds of antifreeze proteins, AFPs. These antifreeze proteins are carried in tandem repeats. And as a result, the fish can actually produce more than one kind of antifreeze protein, which can give it adaptations for different temperatures of water, which allows them to survive better. So in many, many cases in the animal kingdom, mutations of probably DNA replication and or meiosis, or accidents of DNA replication and or meiosis, have produced cells with more than one copy of a gene, which ended up being a bonus to the organisms in some cases because the extra proteins could A, allow them to do more of what those proteins make them do, or B, it allowed them to mutate into different forms for different capabilities. Now, that pretty much concludes our discussion of microemergence.